Have you wondered how food becomes usable energy? Carbohydrates, fats, and proteins are oxidized to harvest electrons and generate ATP for vital work. In this video, we build a foundation, redox principles, the electron transport chain, and oxidative phosphorylation, plus protective mechanisms against harmful oxygen-derived molecules. First, we'll explain oxidation and reduction, and the enzymes that shuttle electrons. Then we'll visualize complexes within mitochondria and how ATP is formed. We'll use animated 3D diagrams of mitochondria, complexes 1 through 4, ubiquinone, cytochrome C, and ATP synthase to make mechanisms intuitive and memorable. Finally, we'll highlight reactive oxygen species, antioxidant defenses, and practical takeaways about efficiency, regulation, and cellular health. Oxidation means loss of electrons, reduction means gain. Biological systems transfer electrons through carriers to conserve energy safely and efficiently. Key electron carriers include NADH and FADH2. They donate high-energy electrons generated during glycolysis, the citric acid cycle, and fatty acid oxidation. Enzymes called dehydrogenases catalyze oxidation-reduction reactions, removing electrons and transferring them to carrier molecules in tightly controlled steps. These electron transfers are coupled to small energy drops, preserving free energy, which the cell later harvests to build ATP rather than losing it as heat. Carriers shuttle electrons to the inner mitochondrial membrane, where the electron transport chain components accept and pass electrons along a defined path. This controlled flow establishes an electrochemical proton gradient across the inner membrane, central to ATP synthesis via chemiosmosis. The electron transport chain consists of 4 large protein complexes and 2 mobile carriers embedded in the inner mitochondrial membrane. Complex 1 accepts electrons from NADH, transfers them to ubiquinone, and pumps protons from the matrix into the inner membrane space. Complex 2 connects FADH2 to the chain, passing electrons to ubiquinone, but it does not pump protons, so it yields less ATP. Ubiquinone carries electrons to Complex 3, which transfers them to cytochrome C, while contributing additional proton pumping to strengthen the gradient. Cytochrome C shuttles single electrons to Complex 4, which transfers electrons to oxygen, the final electron acceptor, forming water. Together, electron flow through complexes 1, 3, and 4 couples to proton pumping, generating a substantial proton motive force across the inner membrane. The proton motive force drives ATP synthase, a rotary enzyme that synthesizes ATP as protons flow back into the matrix through its channel. Each full rotation of ATP synthase produces multiple ATP molecules by combining ADP and inorganic phosphate in the catalytic sites. Chemiosmotic theory links electron transport to ATP production. Without a gradient, ATP synthase cannot produce ATP efficiently. A typical NADH yields about 2.5 ATP and FADH2, about 1.5 ATP, though exact yields vary with organism and conditions. Cells regulate oxidative phosphorylation through substrate availability, allosteric enzyme control, and the balance between ATP demand and proton leak. When coupling is imperfect, some protons leak or electrons escape, reducing efficiency but sometimes generating heat for thermogenesis. A small fraction of electrons can prematurely reduce oxygen, producing reactive oxygen species like superoxide and hydrogen peroxide. Reactive oxygen species can damage lipids, proteins, and DNA if not controlled, contributing to aging and disease when excessive. Cells employ antioxidant defenses. Superoxide dismutase converts superoxide to hydrogen peroxide. Catalase and glutathione peroxidase detoxify hydrogen peroxide. Balanced RO signaling is important. Low levels act as signals for adaptation, while high levels cause oxidative stress and pathology. Key takeaways. Biological oxidation harvests electrons. The electron transport chain creates a proton gradient, and oxidative phosphorylation generates most cellular ATP. Understanding these processes clarifies metabolism, disease mechanisms, and therapeutic strategies. Thanks for watching and keep exploring cellular bioenergetics.